Okay, that was a false start. Looks like it's going now. Just verifying I can hear the audio. Start. Looks like it's going now. Okay. Just verifying I can hear the audio. Start. Looks like it's going now. Okay. Just verifying I can hear the audio. Start. Looks like it's going now. Okay. I'm going to do a quick audio check. This is Stentors Part 2. You can see there's one here that's actually no longer with us. It was already like that in the on the slide when I mounted it from the tube. The specimen selection is getting quite old. I should have done this a couple days ago. So I'm just doing a quick check before I lose all of them, but I may have already done so. Been busy with all kinds of stuff lately, and they were in the fridge for a while, and yeah, so we'll see what happens. They may not be any live ones left. So here's a small one that's similar to the one I showed you last time that lysed. Basically, most of these guys, as they die, they lose the. It's kind of like the sun with fusion versus gravity. These guys lose the water pressure battle because being a high solute concentration inside a semi-permeable membrane, the cell membrane, they are constantly fighting water pressure. So osmosis wants to balance the pressure, and since they're salty inside and the water around them is not, in this case, they're bailing constantly to not pop. Just like you put your red blood cells in fresh water, they will eventually balloon out, lose the little indented center region, turn into a sphere, and basically pop. These guys will as well, so they have, they basically do, their little bailing mechanism is called a contractile vacuole. And you can see them really well in paramecia and some other things, constantly pumping to keep that nice water pressure balance with the environment. So when they die, the pressure stops, getting tweaked by their contractile vacuoles, and then they'll eventually lyse. And there are other reasons they'll do it too, just degradation from age, or I've read there's stuff about temperature and pH messing things up sometimes. So anyway, this is just a quick look at this guy. I think the water's already advanced on him because I waited too long. Let me just do a quick close-up here. Yeah, the water's already kind of gone through and kind of dried up this specimen here. So that's what they look like when the water has advanced. You've seen the wavefront come through before. It's basically the air pushing through as the water evaporates. You get the kind of reverse of an ocean wave going backward as the water goes away, and usually takes things with it and also leaves things behind like this to slowly desiccate. It's still got some moisture in it, but it's not, see, things are all constricted now and bound up into little groups. Anyway, I think the slide may be done. Let me just quickly go around here. Then I'll turn on the music. So let me just check. There's the wavefront right there. See that line? So to the right is air and to the left is water. And water is evaporating. I can see some tiny little guys in there, like tiny, tiny little flagellates, very tiny dots. You probably don't even see at this magnification. By the way, I'm using a new setup tonight. Finally got around to using my Nikon mirrorless camera on the stream. So it may not make a huge difference on the stream itself because I found it a little bit tricky using the Nikon webcam app versus using my little micro software, which had lots of controls in it. This one has less, so I'm having to do more control from the microscope, kind of old school. And so I might have to do a lot more manual tweaks tonight. Anyway, I'm just doing a quick survey before this thing dries up. This wavefront on the one side, something else that looks like it laced a while back, all fragmented. Let's see what else we got. There's the air bubble. Looks like little fluffy clouds on the little margins there. Okay. So nothing is catching my eye here. Anyway, nice thing about this camera, obviously it's a hell of a lot more resolution if I want to go up to 25 megapixels. And also, I get the other goodies like slow-mo, stuff like that. So if I want to do some high quality slow-mo on the stream to slow some of these fast guys down and show you guys some details, that is another advantage. All right. That's an interesting shape there from the polarization of the light from the crystal there. It's a crystalline structure, getting different colors and shading there. In the future, I'm working on a polarization stream as well. There's a cool technique you can use with plane polarizers and actually look at like vitamin C crystals and other kinds of things that are polarized light, fun objects under the scope. There's another polarized object there, polarized by the crystalline structure. Yeah, I'm not seeing anything between the wave fronts here. So I'm going to bail on this slide. Yeah, nothing. Nothing. Bunch of debris from stuff. Okay, well, so much for that slide. All right, well, I'm going to build the slide. Let's start the music. So, as before, JX3D graciously allowed me to stream some of his really cool electronic music on the stream. And so I'm doing it again with a different album. And as last time, I'll give the information here. This is JX3D's website on Bandcamp for this particular album I'm playing tonight. You can go to the main page from here and listen to all of his albums on there, and please buy some. So this is the link for what I'm playing tonight. And as usual, though in there, please to buy some. And so yeah, Bandcamp, JX3D. Henrik will be very happy about this. So I'm going to put the chat up here out of the way now. I don't need that in the way. Sorry, I'm just doing some housekeeping here. Too many windows open. Okay, bear with me while I start the music and balance it. Okay, bear with me while I start the music and balance it. Okay, bear with me while I start the music and balance it. Okay, bear with me while I start the music and Okay, that should be the right volume. I think that's what I had it set for last time. Let me quickly go back and listen to it real quick. Hold on. Volume, I think that's what I had it set for last time. Let me quickly go back and listen to it real quick. Hold on. Volume, I think that's what I had it set for last time. Quickly go back. Okay, that's the best I can tell right now. I'm getting kind of a loop back on it when I try to listen this time, but I'm able to mute that. So I think I'm coming through okay based on the meters here. Just do one quick verification on my phone as a source of truth since it's not connected to the computer. Check 1 2, check 1 2. Sorry about that, got a little close to the mic there, but sounds good on the phone, so I think we're good. Anyway, so where were we? Let's make another slide. See if we can find some existing Stentors here. If you guys also have questions about the music, I know a little bit about Henrik's process. We often appear together on some music podcasts and stuff, chat rooms, music tech chat rooms and stuff. So I have a pretty good idea of what he's using on these tracks, but it's definitely some speculation. So if you have any questions, I can try to answer some of that. All right, let's get a sample. Let's see what we got here. Boy, some dust on these cover slips. I gotta wipe them down. They usually come out pretty clean out of the package, but these are brand new cover slips and they're quite dusty. It's okay though. Do a quick survey. Looking for the big stuff. What is that? Very interesting. Not quite sure what that is, but it's interesting. It's quite small. This is 400 power. Not sure what that is. Interesting structure though. So let's see the 3D structures if focus through it. There's a big depression on one side, looks like the Death Star. And as I focus this way, it has like a little bit of a waist. There's a little groove that runs around this, almost not quite the center of it. I mean, it looks vaguely like the shell or test of a dinoflagellate, but it definitely don't have any in the sample. This is fresh water, so interesting. Just weird stuff to find as I'm poking around. Can also try digital zoom on this a little bit. That's what I do for the Astro streams. It seems to work pretty well. Let's do, that looks pretty good. Let's make sure that's focused correctly. Let's go to this object here. Let's see. Hey Paul, what's up? Enjoy the JX3D music. This is another album of his. Okay, it's holding up pretty well. Use a little bit of a digital zoom since I'm not using the micro software as I usually do. This is my Nikon mirrorless camera running through Nikon webcam utility, which is bare bones as it gets. But it's working out pretty well so far. Just some adjustments to make. So as I said in the stream, I'm looking for Stentors. This is a funny little thing I found in here. Not sure what it is, but it's pretty cool. Definitely better resolution on this camera than the normal camera. Just have to dial it in right. Interesting little structure there. Okay. Where we left off, let's see. Lots of debris like little plant bits. Air bubbles. Sorry, it's a little bit of vibration. That's me shaking the table. If I put too much water on the slide, it tends to slosh around a bit. There's something that's a little bit of a structure to it, but it's not doing anything anymore. Hey Manny, what's up? The chicken story, that's funny. Yeah, I believe it. They're mad scientists out there doing all kinds of crazy shit, so I'm not surprised. But it is amazing, as I was saying in the chat, my mom did decapitate that rooster. It was a real bastard. It was kept attacking me and going after the dogs and stuff. Just kind of a nasty little rooster. It was just like a bully. A little bantam rooster, like the tiniest one. It was a real brat. So mom said, it's just, you know, there's no point to this. I don't think we ended up eating it. I think it was just getting rid of it. But anyway, she decapitated it, put a box over it, got the hatchet out, chopped it off, and then when she took the box off, the body went flying up in the air with a fair amount of precision for something with no head. Tons of energy. It must have flown up about 7 feet. I mean, I was a little kid, so the size is different, but of course it's way off balance because there's no brain to really control it. Oh, there's another one that liced, looks like. See all the vacuoles in there that are sitting there? Those are the vacuoles left behind after the cell liced. So there's another stentor that didn't make it. Some pretty damn hardy vacuoles though. Usually when they lice, those things pop. So it may have just happened while I was searching through the slide. Pretty crazy looking image though. We'll try dark field, I think. Let's see if I can. A little more dark field action on here. There you go. I got it tricky to get the exposure right because sometimes it's too much glowing bits. There we go. That's pretty good. See the vacuoles are still intact. Hey Teeny, what's up? Yeah, watch the other streams, Teeny. The last stream I reposted in my video section because the live one missed the music. I messed up something. So good Stentor action on that one. They're live, although one did die at the end. If I change the light, you can see the particles kind of streaming by here. There's some more oblique lighting there. Gives it more of a 3D pop. Right about there. Thanks, Paul. Yeah, that jam was a totally last-minute affair. Like, I was basically not around on Sunday, and then Sunday night I was just so wiped out. I couldn't even stay up late. I'm usually a late night person, or super late. I think I fell asleep around 7 o'clock or something like that, which is unusual. And slept almost. Well, I got up a couple times to go to the bathroom and stuff, but I slept about 12 hours. So just kind of crazy. And then I woke up and I went, oh shit, I have fractions of time to get this jam done. And then I go, oh, it starts at 1 o'clock. I get a little more time, but it still was a race to get it done because, you know, I can bank something out usually a couple hours without any finesse. But I wanted to add a couple things to it. I thought, well, this needs some Halloween screams and some creepy, you know, stuff in there. So I put in one kind of a specially looped reverse scream. And then I found a nice male and female scream I could interchange during the jam and stuff. And added a couple more percussion bits. I said, okay, that's all I have time for. I got to upload it. So that's what I did. But I have to go back and watch the stream. There's some good ones on there for sure. Mandy was doing the SY99 thing, which is always cool. I got to get mine fixed. I still have that top key to replace, so I can stop playing with it and learn about it more. All right, so we're looking at dead things. Let's keep going here. Oh, that's debris, but it looks interesting. All trailing bits. That's a cool series of air bubbles there. .3D air bubbles. nothing on this thing. It's just debris and air bubbles. How's the music speech balance? Is it okay? It seemed okay on the little test I did on my phone, but it was a very quick test. Yeah. There's nothing there. Dirty ass slides. They're supposed to be clean out of the box, and they weren't. Alright, so much for that. Cool, thanks. Nice to have the verification. I hate to have it screw up like last time where it looked like it was playing JX3D's music and the VU meter was going, but there was a very small mute button that wasn't totally obvious to me. So I'm still learning OBS, and I'm like, oh great. So I went the whole stream with the music muted. So, very silly. Alright, let's try this one. I was trying to save time without setting up both scopes, but I think I should have set up the other one just to use it to find these guys. It was pretty fun last time where I could find them in the petri dish and then suck them up and move them on over. Alright, this time I'm going to clean this cover slip so it's not all dusty. Too much dust. Okay, there's some debris there. Let's go to focus. Plant parts and such. Okay, that's focused. Let's do a sweep. Bubbles. More junk, more air bubbles. Yeah. Looks like another one we have lysed. Or something very similar. They are green, so I'm assuming this is another Stentor that went south. But there's an interesting object next to it. What the hell is that? Fix the, let's do a little dark field first so you can... What is that? Interesting. So you can see all the little dots around the Stentor. There's again the contractile, or just the regular vacuoles that it ejected. Haven't popped yet. But there's something very interesting next to that. What is that? That might be... Oh, that is cool. It's either a very young Stentor, or it's a kind of Vorticella. It's definitely alive. You can see the stuff cycling around it. It's creating a little vortex while it's kind of feeding on stuff. Let me zoom in on it. Oh yeah, look at that. Yeah, it's a young Stentor. So I got one finally. Okay. Let's get the light levels dialed in here. So just get a little more transparency so you can see the guts. See all the vacuoles inside that are all intact? That's what spilled out of the one next to it there, all those little spheres. So this is a small one. The other ones are a little bit bigger than this, as you can see by the big mess over there to the right. There's also a bunch of little guys swimming right next to it. But you can see the striations going left to right in this case. So from what is like essentially the mouth in quotes opening down to the tail, it's a little pointy bit. They're usually fairly conical. And so it has the huge cilia by the opening. Filter, bringing the water in. Bigger cilia than on the body. Body isn't just the cell itself, since it's just one cell, but and there's some food down there for it. All those little dudes cruising around, a little swimmy. Those little dots down there. I gotta change the. So those little dots there on the right, all swimming around. So it's trying to eat those guys, potentially. That's why it's hanging out here. So you can see the cilia going crazy at the opening there. And if you focus at a certain level, you see these long kind of lines, like a striation effect. And that's what I was zooming in on the last stream when that one died. Really cool bundle of almost like a single cell equivalent of muscle fibers, myofibrils. Yeah, I gotta zoom in on this guy. There's all kinds of stuff going on in the cell. The only problem is, since this guy's kind of small, I have to use my less than stellar objective that I had replaced, which has a depth of field issue, but let's try it out. It's contracting because it's not happy. This is that little weird movement they do when they turn into a ball. See the opening where all the cilia are kind of closes up? They're the striations I'm talking about. And so you have a real depth of field issue. I have to keep focusing to show the whole thing. And that kind of section there opens up more and has these long cilia. Now it's extracting itself. It had contracted before. And it's getting bothered again. It keeps contracting. You see that rim? Almost like a giant lip. And it has cilia all around it there. You have to focus through it to get it. You'll see the cilia beating like crazy. Focus through here a little bit. This is not my favorite objective. You can see the cilia there. Focus in it now. It needs better lighting to show it off. And also, there you go. So it's like eyelashes almost. Nice line of, nice grouping of it on the rim there. And you can see there's almost like a whirl, like a spiral whirl of the way the opening works. It kind of has this, looks like it's inside, but these are actually on the outside. See this kind of a whirl arrangement there? It's not just a circular rim of cilia, it's kind of a spiral arrangement, which is why it's really hard to focus on all of it at once, or see it all at once, because it's often on its side. There's a nice shot of a bit of the whirl. There's a giant vacuole there. So there's the whirl and then the spiral bit. Something's bothering again, it's contracting. Now it's a sphere again. This is part of the behavior the guy was studying at the Maker Faire. When they get disturbed and kind of roll up as protection. See the striations there? Look at the lines as I focus through there. They run longitudinally down the cell. And I'll eventually zoom in again with an oil lens so you can really see them. The problem is once I do that, I'd have to clean the cell again. I mean clean the cover slip, and I could lose the specimen. But look at those little markings there. And the opening of the cell as well is so cool. Definitely better resolution than my other camera, that USB camera. So the cilia are beating like crazy on the margins. And because they're out of focus, they're making that margin dance a little bit. See that? You know, the striations in the background. Much like another animal called Blepharisma. It's kind of a reddish color ciliate. It's also very large. And it has these long striations down the body. When I say body, I mean cell, of course. Now I focus through and look at all the vacuoles in there. All kinds of stuff to look at on this guy. And it's this nice green color. I couldn't get the green color to show up on the other USB camera for some reason. It was just, it was making it more dull looking. And it's even more light green than this in the eyepiece, so I can, because I'm zoomed in so much and I'm cranking down the light so you can see the structures better, you're missing some of the colors. So now we're getting more of the green. And it got annoyed again, so it contracted. But I love that section at the top. It's like a giant, like a mouth actually, even though it's not, but it's shaped like a mouth. So they have a section inside there where they draw the critters in with their cilia, and then when they touch the surface of the cell, they make a regular food vacuole. The vesicle buds off the cell membrane, brings the critter inside to digest. And then once they're done with that, you can see in the last stream I did, one of the Stentors is actually dumping his contents like space junk. He brings the vacuole to the membrane and releases it out to the wild. You can see it actually basically its version of taking a dump. It's kind of funny to catch that. I've only caught that a couple times on stream. Sorry, guys. Enthralled by the Stentor. So. Alright, let's focus some more again. I don't have the right config at this magnification to do dark field. I have to do some more tweaks, so I'll just leave it like this for now. There's your little spaceport here. Stuff going in and out. Oh, got disturbed again. It wasn't me, I barely touched the scope. Anyway, the guy's research I was talking about in the last stream, where I got these from. Oh, that's really cool. Look at that detail. They do this contractile behavior for protection, and if you do it repeatedly in a certain time window, at some point they stop reacting because the cell is not getting any damage. It has some kind of a mechanism, so it just costs a lot of energy every time it has to contract, and also it's not feeding. And it's also vulnerable because now it's just a sphere that some bigger animal like an amoeba, or big enough animal like an amoeba, can come along and try to eat if it wants to, or attempt to eat, or some other bigger multicellular animal. So it behooves them to not do that too much for lots of reasons. So they have this mechanism where they stop responding with a certain frequency, and they made a little thing that they could time the disturbances to force it to contract, and figured out this timing scheme. And anyway, so there was that part of the research. The other part of the research was, as he told me at the thing the other day, some new stuff since I talked to him last, is they were trying to figure out if there's a protein encoding or something that acts as kind of a chemical memory to turn the trigger on and off. And they did some testing where it went into they gave it some treatment where basically they stopped the formation of certain proteins, and they got it to not respond anymore, but they also couldn't turn it off. So then it was susceptible to damage when they did that, because it would no longer contract at all, apparently. And I'm probably paraphrasing badly, but I have to go back and read the article. Anyway, and he was trying to see if there's possibly a heritable trait from the offspring. And so far, I don't think that had any evidence or not, but well, it's so cool to see that opening like that. The other ones I couldn't get this angle. It was always like a total side view, but this one's kind of facing me now. So you're really getting a view inside down the barrel, so to speak. But you can see how it's not a perfect little circular area. It's kind of a weird shape because it has this whorl arrangement. It's like two curved surfaces, almost like a Mobius strip, for lack of a better term. So it's really hard to focus on the whole thing and get a whole face-on view. I've got to look for some extra, some electron microscope shots of these guys. Get some nice 3D shots of them. Look at the scanning tunneling kind of deal. So cool. I want to give it more brightness for more of the color, but then I lose the detail. It's more of this kind of light green thing. That's pretty good. That's not bad. It's a little lighter green than that. Here it looks a little more yellow. That's partly the lighting. I'm using a halogen light because it's the problem with it generates more heat than an LED light, which also causes the water to evaporate sooner. But it also gives a more natural, warmer light than the cold-ass LED light. Nice thing is this thing is pretty sedentary. It's not doing much. It's just hanging out. See how those filaments kind of layered, almost like muscle fibers. They move in unison. And it's really cool when you zoom in on them. Alright, we've probably gone through this enough. I'm gonna go, let's go in for the close-up, because who knows how much longer this guy's gonna be happy. So let's do the oil. This will be good. Let me get my lens tissue ready. Just doing a quick pre-clean on the thing, because I'm pretty sure I cleaned it last time when I finished, but sometimes I forget if I'm in a hurry to shut down. Quick, nice wiping of the oil lens. So I can go to 1,000 power. That was 400 power, now we're going to 1,000 power. Let's put some oil on here. Fix that refractive index. And here we go. Dark at first. So there you go. Those are the bundles up close. All these little dots. And then here's that rim. You can see the cilia really well now at high mag. Those are basically the base of the cilia. They're actually going off out of focus here. Let's go back to this inner central area here. See all the bundles in here too. Can I get a... Yeah, I can't do dark field on this guy. To play with it like this. So now it's moving, and now I'm looking through more structures because it's turning. There's a shot of the cilia there. Trying to find the optimal setting for some of these details. Now you can see some of the critters swimming around it pretty close. These little flagellates. Because at this power we're able to look at bacteria pretty well. But these are just little flagellates swimming around in the area. Focus is a really shallow depth of field at this level, but this guy's got some color here. Look at that. Some little red dots there. Might be an actual ice spot at this level. Oh, there you go. That's a nice shot. Looks like a close-up on the dollar bill with the green printing. Thanks, Manny. Thanks for hanging out. Got some nice hot Stentor action here. And I got some damn thing on the optics there I gotta get off. Hello, Rishikesh. Welcome to the stream. This is Stentor, giant ciliate. I got from a new friend who does research on them. You can order from biological supply houses, but I'm lazy, so I was able to get some from him and talk to him about his research and stuff. Look at those fibers, that's so cool. See the individual dots that make them up. Now if I focus way back, you can see the body, the cilia all over the cell. But right now I'm more interested in these filaments. Really interesting. And I saw this in the last one. There's some little pigmented areas that are more green than others. Oh, that's why there's something behind it. Like a vacuole full of yummies. Digesting away, little tiny stomachs. Look at all the activity inside the cell. It's just, you can actually see the cytoskeleton because of the paths that they're taking. They're not totally random. Back to the rim here. That damn particle in there is driving me nuts. Is that on the actual camera? No, it's inside the optic path. Could be on the actual, yeah, it's too small. It's too big to be on the objective, so it's going to be somewhere else. How's this for a screensaver, Paul? Very trippy. Finishing the edge of the cell. Some cilia on the right there. So I have to keep focusing to get through here. Tree rings there. It's pretty wild. Yeah, not outside the cell. It's hard because I want to make a brighter image, but then it gets more washed out detail-wise. I could probably crank it up one more notch with this guy. That's the wrong way. There you go. It's got something blocking it here. I don't know what it's behind. Not sure what's going on there exactly. There's some cilia. Now you can see those striations again. Some cilia on the side. So glad I found some more live Stentors in this. I was afraid I left it in the fridge too long. See if I can get a little more light in here from the camera. And then I can offset the difference here and still keep some brightness. It's not helping that much. Hey Lone, what's up? Yeah, it's Stentor madness again. Music by my friend Henrik, goes under JX3D. And what better time than now to put the link in here. The album I'm playing from him on Bandcamp is right here. And of course, as usual, gotta put this on here too. He's got a lot of really nice electronic music on his channel, his page for Bandcamp. Last stream I did with Stentors, I used a different album. This is, I think, slightly older. I think this is one more before the last one. And he just released a new one. Uh-oh, I'm seeing movement. Did this guy lice? Oh, he did. He's hemorrhaging. Oh well. You know, you live for so long and something happens. Time to go out to a higher mag and see the damage. Or lower mag, I should say. Oh wow, he's already just totally spread out in the area here. Holy moly. That's what happened to him. So the part I was studying was still active and happy, but the rest of the cell had started to lice. And now he's spilled its contents into the surrounding water. All the vacuoles are still intact. You see all those little spheres everywhere, just like this other one here that did it earlier. Their vacuoles are still here hanging out. But this one just liced. And now the material is going all over the place. Let's do a dark field. You can really see the material spread. All that discolored area, it's grayish versus the dark background. That's all the material, all the cytoplasm from the sky. And it looks like the slide is starting to dry up too. Is that the wavefront? Something's happening. Yeah, there's the wavefront. As the air is advancing, the water is receding just in time. So this guy's going to get smushed by the air pocket anyway. Makes kind of a nice art piece there. Like an old Peter Max painting or something from the 60s, 70s. But it's really interesting if I go back to normal view. Look what's happened with the vacuoles here. See, I'm seeing it in the eyepiece. Can I not see it here? There's a rim of of little flagellates and stuff forming around them, like little orbits. See right there, look at that. It's like a halo, it's like little moons going around a planet. All the flagellates that are around here are now having a good old time with all the debris left from the cell lysine dying. So they're forming little arcs around the vacuoles, like little either moons around a planet or accretion disks around a sun. That's wild. Never seen that before. All of these have it. Look at that. Oh, there comes the air bubble over here. So all this cool formation is about ready to go away. Right now I'm in oblique lighting. This is dark field. Oh, still lysine, look at that. More stuff shooting out of there. That's crazy. All kinds of debris getting pumped around. Yeah, it's a metaphor for ours for sure. Sadly, you're right. Everything's falling apart. And now you can see there's the current. Now totally lysed. Look at that. Oh man. Now the big air bubble came in and spread it all apart. That's crazy. And now the air bubbles are going to join as usual. And now we're left with just a few pockets of water here. As always happens. But those vacuoles are some hardy damn vacuoles. They haven't broken yet. A lot of the ciliates I've captured in the past when they lyse and their contents spill out. The vacuoles often pop, sometimes a couple minutes right after they've left the cell, but those things are still going. This is kind of how they reinforce the idea of the endosymbiont theory, where vacuoles and other organelles, mitochondrion and chloroplast and all those kind of things were formed from captured prokaryotes instead of an early forming eukaryotic cell. And they eventually started living together in harmony and like a mutualism, and they eventually differentiated into just individual organelles living inside the cell, which is why mitochondrial DNA is different from the DNA inside the nucleus. That's cool. They're all lined up. So even in death, you can find beauty. Look at that. Let's try non. There's some color there. Typical bright field view. Again, those are some hardy organelles. See, they still have contents. It was digesting things. But now they're just stuck until they decide to pop. Pretty soon the whole slide's going to be dry anyway, so I should pull it out at this point. Make one more slide for the show. One more slide for the show. We're been going on what? almost an hour and a half. I could do another couple slides, I guess. I don't go too long because all I have are these Stentors, nothing else to look at tonight, and I don't want to make the stream too long on one specimen. That was really good though. That last one was amazing. Better than the last stream, I think, as far as getting different angles and lasting longer before I actually split up. Okay, so let's see what else I can get. I think I'm down to my last. I know the guy can only give me so many Stentors from his collection since he was doing a show. So I'm not sure how many more I'm going to find. But we will see what happens. Ah. Too many slips. These cover slips come out all dusty, so I have to wipe them each time now. Kind of annoying for something brand new in a sealed case. Alright, what do we got here? Okay, here's the cover slip. Again, I put too much water on. You can see it floating on top of it, which makes it wiggle, which is not good. Not good. Okay, I'll start with dark field. Why not? Dark field's fun because you can see all the particles dancing, even if they're junk. Okay, I'm just checking the alignment. Well, that's way off. Why is it not showing that in the middle? Interesting. Should be right in the middle of the frame. Oh well. What's that? What the hell is that? Nice little cluster of colorful muck. I zoomed in, I didn't align my zoom on the camera. There we go. Let's get these two to line up. That was dumb. The whole time I was zoomed in at a weird angle. Now they're lined up. Of course, I had an angle now. That's not good. Okay, good. Cool. Just some colorful debris, but there's some little guy swimming next to it. That's kind of nice. Okay, what else can we find? More plant debris. It's a little more dark.